Hi everyone, welcome to our last lecture of the semester. We're going to wrap up the semester with a discussion of a specific division of the nervous system called the autonomic nervous system. Another name for the autonomic nervous system is the visceral motor division. The autonomic nervous system is a really important division of the nervous system. It is the system of the body that is going to help with the regulation of vital life processes. It'll help regulate things like heart rate, blood pressure, body temperature, as well as control things like the fight or flight stress response. The word autonomic means self-governed. And the autonomic nervous system is independent of our own free will. So essentially, the autonomic nervous system is active all the time in help regulating some of the things that just happen automatically within our bodies, like regulation of heart rate or blood pressure or other related activities. So those aren't things that you consciously control, they are unconsciously controlled by the autonomic nervous system. So the autonomic nervous system is really important for helping humans maintain homeostasis. And the scientist who studied the autonomic nervous system in early years was a physician scientist named Walter Cannon. Walter Cannon actually coined the term homeostasis and also the phrase fight or flight. And Walter Cannon actually devoted his entire career to studying the autonomic nervous system. He found a lot of things out about the autonomic nervous system, but one of the most important findings that he discovered was that if you have an animal that doesn't have an autonomic nervous system, they can't survive on their own. They have to be kept in a warm, stress-free environment because they're unable to adapt to changes in the environment. So the autonomic nervous system isn't just important for helping us maintain homeostasis, that relatively constant internal body state, it's also important for helping us adapt to changes in our environment. The autonomic nervous system is a motor division. So what that means is it will actually originate in the central nervous system, and then what it does is it will project out to organs of the body. So the autonomic nervous system actually starts in the central nervous system, but the synapses on the organs, the synapses will actually be out in the periphery on the organs. So once again, this is a motor division. And the motor divisions always start in the central nervous system and then they end in a different structure. So this division does control things like cardiac muscle. More so, it influences the activity of cardiac muscle, but it can control smooth muscle contraction as well as glands of the body. So the autonomic nervous system can innervate things like salivary glands and cause you to produce and release saliva from those structures. Another name for the autonomic nervous system is that visceral motor division. And again, let's break it down. The motor part of this means we're going to be carrying information out of the central nervous system to some specific structures. And those structures are the viscera, or the organs of the body. So this is an output pathway from the brain and spinal cord that allows the brain and spinal cord to regulate organ function. The autonomic nervous system has two main divisions. They are the sympathetic and the parasympathetic divisions. And we'll talk a little bit more about those in a little bit. They do tend to have different or contrasting effects on organs. And we'll talk about why that is. The primary organs of the autonomic nervous system will be the organs that are found in the thoracic cavity. So that would be things like the heart, The bronchi and the bronchioles, those are the tubes that bring air in and out of the lungs. A lot of blood vessels can be controlled by the autonomic nervous system, and also viscera in the abdominal cavity too. So things like your intestines, the stomach, and lots of other structures as well. The kidneys are under autonomic control. So a lot of really important organs are innervated and controlled by the autonomic nervous system to change their function and make sure those organs are doing what they need to do at any given time. The autonomic nervous system also innervates some other structures, things like the blood vessels that you find in the skin. So superficial blood vessels are innervated by the autonomic nervous system, as well as sweat glands that produce sweat. And the erector pili muscle found in the skin is also innervated by the autonomic nervous system. And we'll talk a little bit more about these other other structures that are innervated by the autonomic nervous system because they're also important as well. So as we said, the autonomic nervous system does have two subdivisions. There's a sympathetic division as well as a parasympathetic division. You probably remember that the sympathetic division is our fight or flight essentially system. So it will respond, it'll help us respond to stressors in the environment that could, essentially put us into a position where our lives are in danger. So if you've ever had an adrenaline rush or even a panic attack, that's due to the actions of the sympathetic nervous system. So the sympathetic nervous system is the emergency system. It triggers fight or flight that allows us to fight or flee a stressor. So the example I always give, and I've probably given it before already in our class, if you were trying to cross the street and you were walking through the crosswalk just like you were supposed to, and a car was kind of coming into your path, blowing through the red light. You're not going to stay nice and calm, are you? You're going to go into panic mode. And that panic mode is due to activation of the sympathetic nervous system. And it'll cause your heart to beat faster and harder. You'll start breathing quicker. And the idea is that, we're trying to pump blood to our muscles and brain, and by breathing harder and faster, we're trying to bring in more oxygen, expel more carbon dioxide to make sure we're getting that oxygen to our muscles and our brain so we can flee that stressor very, very quickly. So the sympathetic division is responsible for that emergency response. It is a survival mechanism that the body has to deal with things that are potentially harmful or even fatal. The sympathetic nervous system does have some important functions even during resting conditions. So that's what the second bullet point is referring to. The sympathetic nervous system isn't just the fight or flight system of the body. It's also really important during resting conditions for helping us to maintain the muscle tone in our blood vessel walls. So what that will do is it will help us maintain blood pressure. So it'll keep the muscle in the blood vessels partially contracted. And what that'll do is it'll let the blood vessels withstand the force of the blood moving through the blood vessel and help us maintain a normal blood pressure. So even at rest, the sympathetic nervous system is active in helping us maintain the muscle tone in our blood vessels, which is important for blood pressure regulation. The sympathetic nervous system uses two different neurotransmitters or hormones. They can often act as hormones too. They are called norepinephrine and epinephrine. You might have heard of epinephrine as adrenaline. That's another name for epinephrine is adrenaline. And a related compound is norepinephrine or noradrenaline. And interestingly, I'll tell you that, Most people know about adrenaline. Okay, that's kind of the one we all hear about, but it's actually noradrenaline that tends to affect most of the organs in the body. So interestingly, noradrenaline can have a bigger effect at times than adrenaline does. And we'll talk about why that is. All right. Um, the sympathetic nervous system does maintain the normal functioning of what are called doubly innervated autonomic effectors. And I want to break that down because there might be some new words in there for us. So effectors are structures essentially affected by a division of the nervous system. And that, just for a few examples, you have for autonomic effectors, things like the heart or the lungs, kidneys, and more. So most of the organs in the body are autonomic effectors. And the organs of the body, like the heart, for example, are what are called doubly innervated. So I think we know what innervated means. Innervated just means that a particular structure has the nerve supply. If something is doubly innervated, it's innervated by both of the divisions of the autonomic nervous system. So just to give you an example, your heart is doubly innervated because it's actually got a nerve supply from both the sympathetic as well as the parasympathetic division of the autonomic nervous system. So doubly innervated just means that something is controlled by both of the divisions of the autonomic nervous system. Okay, so we will talk more about the sympathetic division here in a little bit. It is a really important one for a variety of reasons. Now, the more active, I would say, division of the autonomic nervous system is the second one. It's called the parasympathetic division. And this is our rest and digest system. Sometimes you'll see it as breathe and feed. Either one is fine. The idea being that for the parasympathetic nervous system, it's going to be most active in times of rest and when you're able to do digestive activities. And for most of us, we are predominantly under parasympathetic control, our organs are, because generally we're in a resting and a digesting state most of the time. If you started exercising, you would shift that and be more in a sympathetic kind of pattern of biological activity. But during most resting conditions, we're largely under parasympathetic control. So the parasympathetic division is actually the dominant controller. It means it's the prominent controller of most organs in the body. So that means that largely we are under rest and digest mode. So hopefully nobody's, having a constant adrenaline rush happen all the time. It's important to have that rest and digest activity. Now, the parasympathetic division actually uses a different chemical than the sympathetic division, and that's acetylcholine. Acetylcholine is a substance that slows heart rate, and it can do things like promote digestion as well as elimination. So acetylcholine will work to promote digestive activities, urine production, and those kind of biological activities that you would do when you're in a resting mode. Of course, if you're in an adrenaline rush or fight or flight, digesting your food or making urine is not really that important because under that fight or flight response, you're trying not to die, So who cares if you can digest your cheeseburger or make a little bit more urine? None of those things are important if your life is in danger. So the body's really good at prioritizing what needs to be done functionally, and it is a balance between sympathetic and parasympathetic activity. Neither division is ever off completely. It's just that the balance shifts if you went into, for example, a fight or flight response. During rest, if this were an activity chart, maybe your parasympathetic division is here and sympathetic is down here, okay? But this would be your 0 activity level. Now, if let's just say you had the situation come up that I was talking about earlier, trying to cross the street, those activity patterns might shift. You might go down to a very low parasympathetic level, but your sympathetic nervous system would take off and become super active as you try to deal with that stressor that could be potentially harmful or even fatal to you. So neither division is ever off. It is a balance of activity. Anatomically, the autonomic nervous system is more complicated than our other motor division, which was the somatic motor division. You probably remember the somatic motor division. It's the division of the nervous system that controls skeletal muscle contraction. And for skeletal muscle contraction, if you recall, we had, I'm going to draw over here. So let's just say we had the spinal cord. So there's the spinal cord, gray matter in the middle, okay? And you had, I don't have enough space here, you had a muscle over here, okay? Okay, so that is, I'm just going to put an M there so you know that that's supposed to be a muscle. Essentially what we have is one somatic motor neuron whose cell body is in the gray matter of the spinal cord, whose axon leaves the ventral root because that's the motor root. Okay, so it leaves the ventral root and it stretches all the way to the muscle. And then we've got a synapse and the neuromuscular junction. Okay, so circled there in blue, NMJ, neuromuscular junction. So that's pretty much just one neuron that stretches from the central nervous system to the skeletal muscle. The autonomic nervous system is a little different because we still start in the CNS, so this is the spinal cord, but instead of having just one neuron that it takes to stretch to the organ, like the heart or the lungs, we have two, okay? So in the autonomic nervous system, there are actually two neurons that it takes to essentially get to the organ. And the first one is a neuron called the preganglionic neuron. The preganglionic neuron has a cell body in the CNS. So it's the one here in green. That is the cell body of the preganglionic neuron. The axon of that neuron leaves the spinal cord here via that ventral root, and it actually terminates in a structure called an autonomic ganglion. Now let's break that down. So autonomic is referring to autonomic nervous system. A ganglion, remember, is a group of cell bodies of neurons in the peripheral nervous system. Because remember, the autonomic nervous system is a division of the peripheral nervous system. Go back and look at that neuro-organizational chart if you need to. But anyways, so the autonomic ganglion is composed of cell bodies of neurons. And those cell bodies actually belong to our second class of neuron. It's called the postganglionic neuron. The circle is the cell body here. And there's a synapse. So I'll circle this synapse here. And that synapse is between the preganglionic neuron and the postganglionic neuron. And it is a chemical synapse. So we'll talk about it a little bit more later, but that synapse between the pre and the postganglionic neuron is a chemical synapse. The axon of your postganglionic neuron leaves the autonomic ganglion, and then it would go to the end organ where we would have another chemical synapse between the postganglionic neuron and an end organ. Okay, so I'll just draw the heart here. And then, you'd have that chemical synapse here between the postganglionic neuron and the end organ. So essentially what we have here is a motor division. It's just that in this motor division, you have two different neurons in the chain. So you've got the preganglionic neuron that starts in the CNS and it ends in the autonomic ganglion. And then the postganglionic neuron starts in the autonomic ganglion and it ends near the end organ. And again, the end organ is going to be something like the heart, the lungs, a blood vessel, one of the organs or other structures of the body. So it's a two neuron chain for the autonomic nervous system's anatomy. And that is something that's important to know, okay? And those are all going to be chemical synapses, regardless of division, okay? So sympathetic and parasympathetic divisions both use chemical synapses at these synapses of the autonomic nervous system. So your two synapses are going to be at the autonomic ganglion between the preganglionic and the postganglionic neuron. And then your second synapse is going to be between the postganglionic neuron and the end organ. So it would happen out in the periphery near the organ. Okay. So a little bit more about this. We've got, like I said, a two neuron chain that allows us to carry signals out of the central nervous system to our organs. And the two neurons in the chain are both part of the efferent or the motor pathway to the organ. And we have a preganglionic as well as a postganglionic neuron. The preganglionic will actually conduct impulses from the central nervous system to the autonomic ganglion. The postganglionic neuron is still a motor neuron, but it starts out in the autonomic ganglion and it actually stretches to the organ. Okay, so it's a chain, right? It's just that this particular chain has two neurons as opposed to the somatic motor division, which only uses the one motor neuron to get to the muscle. Okay, I like this picture because it shows us a comparison of what I'm trying to say in words, but sometimes a picture is a little bit easier to process. So the top part of this diagram is showing us the somatic motor division. So they put efferent, but that just means motor. So this is the somatic motor division and how it is able to innervate muscles. So this is a skeletal muscle. The skeletal muscles are somatic effectors. Remember, somatic means skin and skeletal muscle. So this green neuron, actually, it starts in the gray matter of the spinal cord, and then its axon leaves the ventral roots, and then you can see here, maybe I'll use a different color so you can see that. That would be smart. Okay. So I'll circle that again in red. That is my somatic motor neuron cell body, and this is a lower motor neuron. The cell body is in the gray matter of the spinal cord, but the axon of that neuron leaves the spinal cord by way of the ventral nerve root. You can see the axon here, and there's no break, okay? The axon stretches all the way to the muscle, and then you've got your synaptic knobs with acetylcholine that we can release onto the muscle to produce a contraction. So that's what we learned in the muscular system. We learned a little bit about this kind of architecture, and really what we had focused on was this, right? We focused on the end of that and talked about the neuromuscular junction, because this would essentially be our neuromuscular junction, and then all the events happening in the muscle cell. Okay, but we've just got one neuron here in the somatic motor innervation that starts out in the CNS and stretches to the skeletal muscles. Compare that to this. This is our autonomic motor innervation. So this is not somatic motor, it is visceral motor. That's our visceral motor division. And make a note here, we've still got our motor neuron, our first one with its cell body in the gray matter of the spinal cord, and an axon of this autonomic motor neuron leaving the spinal cord, still through the ventral nerve root. And the axon stretches to the autonomic ganglion where it synapses on a postganglionic neuron. So we've actually got a synapse here. We're not at the organ yet. Okay, we'll get there, but we need another neuron to pick up the signal. It's like a relay race, and this neuron, number one, the preganglionic neuron we just talked about, is passing the baton on to the second neuron in the chain, which is the postganglionic neuron. And it will be that postganglionic neuron that takes the signal the rest of the way to the end organ. And this is a chemical synapse, okay? And it's always going to be acetylcholine at this preganglionic, postganglionic synapse. So this preganglionic neuron, and I'll go ahead and label this. So this is the preganglionic neuron, okay? That one is always going to release acetylcholine. It's never different. It's always acetylcholine there. The acetylcholine actually stimulates the postganglionic neuron, who fires an action potential that is conducted down the axon, and then that triggers exocytosis of either acetylcholine or norepinephrine onto your end organ. And I'll make a notation here that if this chain, okay, if this was the parasympathetic nervous system, we would be releasing acetylcholine onto the end organ. So this would be if we were releasing, if this were a parasympathetic pathway, the neurotransmitter released would be essentially acetylcholine. If this were a sympathetic nervous system motor pathway, we would be releasing norepinephrine onto the end organ. So the neurotransmitter at the end of the chain is different depending on which division of the autonomic nervous system it is. But both the sympathetic and parasympathetic nervous systems are laid out this way. We have a starting point in the CNS, and then we've got neuron number 1 and then neuron number 2 to get to the end organ, okay? Okay, I want to take a look at the anatomy of the parasympathetic nervous system and show you the location of the pre and the postganglionic neurons in the parasympathetic nervous system. In the parasympathetic nervous system, the preganglionic neurons their cell bodies are located in two places. They're located in the brainstem, as well as the sacral spinal cord. So for the parasympathetic nervous system, the preganglionic cell bodies start out in the brainstem or the sacral spinal cord. And then these long stringy things, those are the axons of those preganglionic neurons. You probably noticed that those green neurons are pretty long. And it is definitely the case that for the parasympathetic nervous system, the axons of the preganglionic neurons are really long. So they are really long and they almost touch the organ actually. So probably the easiest one to see that trend is if you look at the lungs. So you can see that green neuron almost touching the red neuron, and the red neuron in this case is the postganglionic neuron. So I'm going to try to write that pre, okay? So all these green ones are preganglionic neurons, and then the red ones are all postganglionic neurons. I'm just going to write post here, okay, just so you can kind of see where those are. And if you look very closely, you can see the dot of the postganglionic neuron. That's the cell body or the soma. So your autonomic ganglion is right here, okay? And it's pretty much, because it's pretty much on the organ, the autonomic ganglion in the parasympathetic nervous system is actually called the terminal ganglion. And that is us getting a little bit more specific than just calling it an autonomic ganglion. It's more specifically called a terminal ganglion because it's found almost at the end of the chain. Terminal means end of something. So, for the terminal ganglia, they're almost at the end of this two neuron chain. They're pretty much on the target organ, like the lungs or the heart, etc. So, anyways, the postganglionic neuron, because its cell body is usually on the organ that it controls, the axons are really short. So for the postganglionic neuron, the axons, okay, or the fibers, are going to be really short. So look at the length of these little red postganglionic neurons. They're tiny, they're short, because they're pretty much on the organ that they control. Now, the parasympathetic division actually uses acetylcholine as the neurotransmitter at both synapses. And remember, you've got synapses between the pre and the post ganglionic neuron, as well as the postganglionic neuron in the organ. So those are the locations of your two different synapses, and it's actually, I'm going to write an A here, it's actually acetylcholine for both of those situations, actually. So acetylcholine is used at both of those synapses. Another important thing, you will notice that we've got some cranial nerves on here. We talked about these cranial nerves last time, and I told you at least a couple of these do have autonomic fibers, including the vagus nerve and the glossopharyngeal nerve. And for the most part, these cranial nerves, they're mixed nerves, except for oculomotor, because they have autonomic fibers. So they are not just like somatic motor neurons, nerve fibers, they are actually visceral motor too. So, these cranial nerves all possess, parasympathetic axons, essentially. So they do contain parasympathetic axons, meaning that we have autonomic regulation of structures such as the pupil, salivary glands, and for the vagus nerve, virtually all the organs in the thorax and the abdomen and the pelvis too. So, these are very, very important things to remember, especially the vagus nerve. I'm going to circle, the vagus nerve here and show you how many branches it has to control organs like the heart, the lungs, and then all these digestive and also urinary organs as well. I mean, it's truly impressive there. Okay. This is my version of the anatomy of the parasympathetic nervous system. And I will say that I'm sorry, I actually did reverse the color on this chart. And in this one, the green is the preganglionic neuron and the red is the postganglionic. So don't let that throw you too much. You could write this out in your own colors if you want. But what I did is, I know that last chart is very cumbersome, and I don't want you to remember everything that's on there. This is not a 3000 level or 5000 level anatomy class, it's a 1000. So I want you to know the basic architecture of the parasympathetic nervous system's anatomy. And so what that means is understanding the location of the preganglionic neuron cell bodies, the location, general location of the preganglionic axons, the location of the postganglionic cell bodies and axons, and some of the fundamental characteristics associated with those. So let's kind of start at the beginning and see what we have. So in the preganglionic cell body anatomy category here for the parasympathetic division, we have cell bodies in both the brainstem as well as the spinal cord. So that was reflected on the last diagram. I just boiled it down on this slide into one flow chart or one slide. The axons of those neurons are either going to be part of the cranial nerves. So our brainstem parasympathetic neurons, their axons join up with those cranial nerves that we were talking about before. And for the sacral level of the spinal cord, the preganglionic cell bodies are in S2 through S4, while the axons actually join up with that nerve that I should have pointed out on the last slide. It's called the pelvic splanchnic nerve. And both of them, okay, whether you're, from the brainstem or from the sacral spinal cord, you're going to terminate or have a synapse. Okay, so there will be a synapse here between the preganglionic neuron and the postganglionic cell body. And that is going to occur within the terminal ganglia. The terminal ganglia are often on the wall of the target organ. The axons of the preganglionic neurons are very short, and they are going to terminate on your end organ. And again, that could be the heart, it could be the intestines, it could be the lungs or salivary glands, what have you. So it's one of the organs of the body. And then, there's your second synapse. Okay, so this is also a synapse. And the parasympathetic nervous system is definitely easier than the sympathetic. Anatomically, it's simpler. And in terms of remembering what neurotransmitters are at each synapse, it's also easier. So at the pre-post synapse, this is acetylcholine. At the postganglionic organ synapse, we also have acetylcholine. So we've got acetylcholine here, as well as here. Okay, so acetylcholine is used at both of these synapses in the parasympathetic division. That brings us to our sympathetic division of the autonomic nervous system. And without a doubt, the sympathetic nervous system is anatomically more complicated than the parasympathetic division. The starting location within the central nervous system for your sympathetic division is the thoracolumbar spinal cord. So the thoracolumbar spinal cord is our origin point for the sympathetic nervous system. The cell bodies of those green preganglionic neurons are actually in T1 through L2 of the spinal cord. And then the axons of those neurons leave the spinal cord by way of the ventral root. And then if you look very carefully at this diagram, you can see those axons leaving, and then they hit this yellow structure with a bunch of essentially kind of chain-like structure all linked together. This structure is called the sympathetic chain. And the sympathetic chain is essentially one of the autonomic ganglia. It is called a chain because the neurons kind of are all linked together in a chain like you can see in the diagram. It is not the only autonomic ganglia that exist in the sympathetic nervous system, but it is the first one that these neurons will encounter. So I want to erase what I circled there just so we have a clean slate to work from. But for these sympathetic neurons, they start out in the gray matter of the spinal cord in T1 through L2, and then the axons leave the spinal cord via the ventral root, and then they hit this sympathetic chain, which is one of the groups of autonomic ganglia in the sympathetic nervous system. And from there, one of three things can happen. We can have a synapse at the same level of the chain as the spinal cord level that we started. Another thing that could happen is that that preganglionic neuron hits the chain and then moves up the chain or even down the chain. And that is, I'll try to draw it this way, okay? That's our second possibility. A third possibility is that our preganglionic sympathetic neuron leaves the spinal cord, hits the chain, and keeps going through without a synapse. And that would be our third scenario, where the preganglionic sympathetic neuron, its axon leaves the spinal cord, hits the sympathetic chain, but doesn't stop. It doesn't synapse. It just keeps passing through and synapses on a second class of autonomic ganglia called the prevertebral ganglia. And for the sympathetic nervous system, all of these are, let me use a different color. All of these are prevertebral ganglia. So all these ones with specific names like celiac ganglion, inferior mesenteric, superior mesenteric ganglia. Those are called prevertebral ganglia of the sympathetic nervous system. So for the sympathetic nervous system, you do have two groups of autonomic ganglia, the ones in the sympathetic chain or the prevertebral ganglia. So let me write that. So for the sympathetic nervous system, for the autonomic ganglia, they can belong to either the sympathetic chain or prevertebral ganglia. Okay, so if we're in that third scenario where we pass through the sympathetic chain and synapse in one of these prevertebral ganglia, you can see the synapse here, the axon of the postganglionic neuron would leave the prevertebral ganglia and then go and synapse on the organ. One thing that you'll note is that for the sympathetic nervous system, those preganglionic axons are short, but the postganglionics are long. So you can just kind of take a look at this diagram and see those different proportions to the lengths of the axons in the sympathetic division compared to the parasympathetic division. That is a point of divergence anatomically. What is also different is that for the sympathetic division, we use different neurotransmitters at our synapses. So at the post-ganglionic synapse, we're still going to use acetylcholine. So that is the same. But at the post-ganglionic organ synapse, we're mostly going to use norepinephrine as our neurotransmitter. So keep that in mind. We do use some different neurotransmitters here. And we will talk a little bit about the adrenal medulla at some point. It is part of the adrenal gland, so it's right here. And the cells in the adrenal medulla pretty much act like modified sympathetic postganglionic neurons. So they can receive acetylcholine via the splanchnic nerve here, and then they can respond by releasing norepinephrine or epinephrine into the bloodstream, where those can actually work as hormones instead of neurotransmitters. This is another diagram showing us a little bit more of a zoomed in view, especially of the preganglionic neurons of the sympathetic nervous system. And you can see those cell bodies of the preganglionic neurons in the gray matter of the spinal cord and the axons leaving the ventral root of the spinal cord. So remember, we're T1 through L2. And then one of three things can happen from there, guys. So this is back to our three possible scenarios. You can have the axon that synapses at the same level of the spinal cord. So that would be a segmental synapse. So if you are a T1 sympathetic neuron, you would synapse in that sympathetic chain. So this is the chain at the same level. So this would be T1, the T1 level of the spinal cords over here. So let's say this were T1, this would be the T1 sympathetic chain ganglion. You can also move up or down the chain. So let me erase. You can also move up or down the chain. So kind of chase this green neuron here that's moving up, okay? This guy is actually synapsing at a different level of the sympathetic chain. So that's our second possibility for what can happen with the axons of our preganglionic sympathetic neurons. The third scenario is this one. Follow this green neuron who just passes through the chain but doesn't synapse and instead goes to synapse here. And this is actually one of the prevertebral ganglia. It is also called a collateral ganglion because we don't have enough terms. But if you see collateral ganglion, just to tell you, if you read it in the book, that's also a prevertebral ganglia. So what I want to do is do a flow chart here, just like we did for parasympathetic, just so you can see it without a lot of these different branches and details. What do we really want to know about this? So as we looked at for the parasympathetic nervous system, I drew a flow chart for sympathetic as well. And this one, because there are more options for the route that the preganglionic neuron axons can take, the flow chart is actually on 2 PowerPoint slides this time. So the one that you're looking at is actually just the preganglionic neuron anatomy and locations for the various preganglionic structures in the sympathetic nervous system. So you'll notice that we've got the preganglionic cell bodies of the sympathetic neurons. They are going to be located in T1 through L2 or L3 of the spinal cord in the lateral horn. The axons of those neurons will leave the spinal cord via the ventral roots and go into a structure called the white ramus. A ramus in the nervous system is a branch or connecting structure. But the axon will be diverted into the sympathetic chain. The sympathetic chain is also called the paravertebral ganglia because they run parallel to the spinal cord in a chain. Once the axons of those preganglionic sympathetic neurons hit the sympathetic chain, one of 3 things can happen. So there are 3 possibilities for the route that those axons will take. The neuron can synapse segmentally within the paravertebral ganglia, and segmental just means at the same level. So if this were a T1 sympathetic preganglionic neuron, it would synapse in the sympathetic chain also at the T1 level. The axon could also move up or down the sympathetic chain or trunk, and that would be the case where maybe you have a T1 sympathetic neuron and it goes down to T5. Okay, so to the T5 level. So essentially you're moving up or down the chain and then there would be a synapse. You could also have those axons not synapse in the sympathetic chain at all, and instead they just pass through and synapse in one of the prevertebral ganglia. And some examples there would be like the celiac ganglion or superior or inferior mesenteric. So this would be the synaptic knob at this this green triangle. And what we would release from the sympathetic knob of our preganglionic sympathetic neurons would actually be acetylcholine. So I'm going to put an A there for acetylcholine. And the acetylcholine from the preganglionic sympathetic neuron would bind to receptors on the postganglionic sympathetic neuron. So once again, just kind of showing you that overflow. So we've got the acetylcholine being released, and it would stimulate the postganglionic sympathetic neurons, which are located either in the sympathetic chain, which is also called the paravertebral ganglia, or within the prevertebral ganglia, like the celiac or superior and inferior mesenteric ganglia. The axons of those neurons will leave the ganglia and go to the gray rami. Gray rami are unmyelinated, the white ones are myelinated. So not a huge and really important difference for us, but those axons will be diverted through another connecting structure. The axons are long, and then they will actually stretch to the end organ, which could be the heart or lungs, etc. And the neurotransmitter at this synapse is norepinephrine. Okay, so norepinephrine is our neurotransmitter at that second synapse in the sympathetic nervous system. Okay, so let's talk a little bit about some of the physiology of the autonomic nervous system. The autonomic nervous system plays a really important role in reflexes called visceral reflexes. We talked about somatic reflexes in prior lectures, and the example that we went over was the knee-jerk reflex. But the autonomic nervous system participates in its own reflexes that are really, really super important for helping us maintain homeostasis. So the autonomic nervous system will help us regulate our organs through these visceral reflex mechanisms. So just like with somatic reflexes, you're going to have an afferent and an integration and an efferent pathway. So I'll just kind of circle and show you what we have going on in a visceral reflex arc. All of these blue areas, and I'll go ahead and circle in blue because that would make more sense. The blue areas of the visceral reflex arc are actually the sensory part of the reflex pathway. So we're either detecting a stimulus or we're conducting that information about organ conditions or organ function to the central nervous system for processing. So all the stuff there in blue is part of the sensory pathway. This green area is actually the integration step. And the integration step is going to happen in the central nervous system typically. So it's typically going to happen somewhere in the CNS because the brain and spinal cord are integrators, right? So that integration step will take place generally somewhere within the central nervous system, although there are some exceptions to that when it comes to like the digestive system. But don't worry too much about those special cases. If you know that the integration step's going to happen in the central nervous system, that's good enough for me. The red part of the pathway here is actually the autonomic part of the pathway, and that's going to involve our autonomic nervous system. So really in a visceral reflex arc, you're only going to have the autonomic nervous system being the red part of the pathway because it's the motor output from the central nervous system. It doesn't really do the integration step as much or any of the sensory part of the pathway, but it will be involved in carrying out the change in that organ's function. So just to kind of point out some of those different aspects of the visceral reflex loop. So I want to go over it in a little bit more detail now. The visceral reflexes are unconscious automatic responses to stimulation that involve visceral receptors and effectors. In a visceral reflex arc, there are structures called receptors that are involved in detecting changes in organ function. And that change could be something like stretch or tissue damage, different chemicals, temperature, and other internal stimuli. So the example that I will give here is blood pressure. detection. So blood pressure is detected by receptors that are sensitive to stretch, because in instances of high blood pressure, there's a lot of stretch occurring in some of the arteries of the body, and we'll get more specific in a minute. So you have a receptor in a blood vessel, for example, that detects the high stretch associated with high blood pressure, because pressure generates stretching. That information about high stretch is detected by the receptor in a blood vessel, and the information about the high stretch will be carried via the axon of what is called a visceral sensory neuron. This is not an autonomic nervous system neuron, it's a visceral sensory neuron. But we'll conduct that signal about high stretch to the central nervous system, who can then process that information. So we'll do an integration step. And for reflexes, they're a predictable response to a stimulus. So if the central nervous system detects high blood pressure and high stretch, it's going to take steps to try and reverse that and get us back down to homeostasis. So what will happen is that there will be an integration step that happens in the central nervous system, and we'll be taking action to try and do things that will lower blood pressure. And so the signal, the motor signal, will go out through an autonomic pathway, and that's kind of the red part of this pathway. So I'll go ahead and erase a little bit here so we can draw that in. So in this red part of the pathway, this is going to be the autonomic output. And if you had to think about it, and I just want you to sit and think for a second here, what division of the autonomic nervous system do you think would lower blood pressure? If you just had to think about it, is your blood pressure going to be lower in fight or flight or during rest and digest? If you say rest and digest, you would be correct. And so this pathway out of the central nervous system is going to occur by way of the parasympathetic nervous system in this example. Okay, so in this example, our autonomic neuron would actually be a parasympathetic neuron. So we have the signal that's carried out of the spinal cord, or actually in the case of blood pressure, it's going to be one of our brainstem neurons. But anyways, so you have your parasympathetic output from the CNS, and that dark red neuron is the preganglionic. So I'll make a note of that. So this dark red one is the ganglionic parasympathetic neuron, and then this paler one is the postganglionic parasympathetic neuron, and that is going to terminate in a visceral effector. So the axon will actually end on a visceral effector, and in the case of blood pressure, that effector is going to be the heart. And what will happen is that we'll release acetylcholine onto the heart, which will bind to receptors on the heart and slow down heart rate. And if we slow down heart rate, we're going to pump less blood into the blood vessel and decrease the pressure. So these visceral reflexes are extraordinarily important for helping us maintain homeostasis and letting us respond to changes by trying to get us back to normal conditions. Now, keep in mind, the autonomic nervous system is only the efferent part of the pathway. It's only this red part of the pathway involving the path from the central nervous system out to the organ. This is a more complete view of what will happen in the instances of a blood pressure deviation. So this is just kind of filling in some of those details to give you more information. If you have to take AMP2, you'll learn a little bit more about some of the things that are involved in blood pressure regulation, but this is a good, companion to what we were just talking about. It gives us some more information. So the reflex that is involved in blood pressure regulation is something called a baro reflex, and baro is referring to pressure. So really, this is a pressure reflex. And what happens is in a few of the arteries very close to the heart, so this is the heart, they actually have receptors called baroreceptors. So we're still following our schematic that we were talking about last time with the receptors and the visceral sensory integration and then the motor pathway, but we're filling in the details and saying what those specific structures are. So for the baroreflex, our receptors are called baroreceptors because they detect stretch. The baroreceptors detect stretch, and there is a visceral sensory pathway, that would be number 2, that will carry information about increased stretch in the blood vessel to the brain by way of the glossopharyngeal nerve. So one of our cranial nerves, the glossopharyngeal nerve, can actually conduct that signal to the medulla oblongata. And then don't forget, the medulla has both a cardiac and a vasomotor center. So the medulla will receive that information. And then here in green, you see this little interneuron that we have. That's going to be your integration step. And the interneuron in the medulla is going to say, hey, stretch is too high. We need to take steps to lower it. And one of the ways that we can do that is by decreasing blood pressure. So this is your third step, the integration step. Looks like I didn't put it in the list here, but the integration step would be right here. And they didn't really label it in the diagram either, so I guess I won't number the integration step, but just remember that it is going to happen there in the medulla. The medulla is then going to, by using parasympathetic pathways via the vagus nerve, it will conduct that signal via visceral motor or autonomic, and more specifically parasympathetic nervous system pathways. We will conduct that signal to the heart, and this first green neuron, that's the preganglionic parasympathetic neuron. And then you can see that we've got the postganglionic one sitting right on the heart. And what that will do is once the preganglionic parasympathetic neuron passes the message on to the postganglionic parasympathetic neuron, the postganglionic parasympathetic neuron will release acetylcholine onto the heart's pacemaker, slow down heart rate, and by slowing down heart rate, we will reduce blood pressure. And your body's doing this kind of stuff all the time, and all of it is done to help you maintain homeostasis, relatively normal internal body conditions. So this is a negative feedback loop. It's just a very sophisticated one designed to help us maintain normal blood pressure. One of the questions that's important to consider is how the sympathetic and parasympathetic nervous systems have different effects. And there are a couple of different answers to that question. One of the big things that causes the sympathetic and parasympathetic divisions to have different effects on the same organ is that, yes, they do release different neurotransmitters onto the organs. So even though the heart is innervated by the sympathetic and parasympathetic division, those two divisions release different chemicals onto the heart. The sympathetic nervous system releases norepinephrine onto the heart, whereas the parasympathetic nervous system releases acetylcholine. And fundamentally, those two chemicals will have different effects on the heart. So, and if you have to take AMP2 and you take it with me, we actually do a lab where we put norepinephrine and acetylcholine onto a frog heart that's still beating, and you get to see the different effects of those chemicals on heart function. It's actually pretty neat. Another reason that the sympathetic and parasympathetic divisions have different effects is that the receptors on target cells can vary. So some target cells will respond to the same neurotransmitter differently because they have a different type of receptor. And a good example of that is one that we're going to talk about here in a minute. But one thing that you might have crossed your mind over the course of these lectures is that acetylcholine in the neuromuscular junction stimulates muscle contraction, right? It's a stimulatory chemical. So then how does it cause, an inhibitory effect or slow down the heart? I mean, those are two very different effects. And the answer is, well, acetylcholine is acetylcholine. But what is different is that in the neuromuscular junction, there's a different receptor. It's a different acetylcholine receptor than the one that the heart has. So the chemical might be the same, but because it's a different receptor, you can have a very different effect. And that's something that we'll talk about here in a few minutes. So there are different types of receptors for acetylcholine and norepinephrine. So the receptor type matters too. What does it bind to? So acetylcholine is an important neurotransmitter. We've talked about it a lot this semester. It is a neurotransmitter that is found at both of the sympathetic and parasympathetic pre-post synapses. So the pre and the post-ganglionic synapses for both the sympathetic and parasympathetic divisions both release acetylcholine. And then the postganglionic parasympathetic neurons also release acetylcholine. So I like this chart on the right. It shows you the parasympathetic and the sympathetic divisions, the preganglionic neurons, and then also the postganglionic neurons. Okay. And one thing to notice here is that for each of the pre and the post synapses, okay, it's going to be acetylcholine at each of those for both the parasympathetic and the sympathetic divisions. And it's only going to be the postganglionic end organ synapse that has different chemicals. So if you look at the parasympathetic division, this postganglionic neuron releases acetylcholine onto the organ, whereas in the sympathetic nervous system, our postganglionic neuron releases norepinephrine. So this is really our big difference there. Okay, so I do like that they show this diagram with all the detail of kind of adding in where those neurotransmitters are in which synapses. So acetylcholine is found at the pre-post synapse for both the parasympathetic and sympathetic divisions, as well as the parasympathetic postganglionic, the parasympathetic postganglionic organ synapse, which is right here. Any axon that releases acetylcholine is called a cholinergic fiber, and any receptor that binds acetylcholine is called a cholinergic receptor. So the cholinergic is referring to acetylcholine. There are two types of cholinergic receptors, so now we're going to kind of focus on those acetylcholine receptors. And they are the muscarinic receptors and the nicotinic receptors. So those are our two main types of acetylcholine receptors. The muscarinic receptors are metabotropic. That means that they use second messenger systems to carry out their functions. And for the acetylcholine, the muscarinic acetylcholine receptors, a lot of these are inhibitory. So this is actually the type of receptor that you have on the heart and a lot of smooth muscle and gland cells as well. So there are many different muscarinic receptors, but the M1, the M3, and the M5 are all going to be ones that produce inhibitory effects or, kind of diminish the activity in that tissue. So the heart has the M1, 3, and 5 receptors. And there is kind of a diagram on the right here, just showing us some of the differences between, for example, a muscarinic receptor and the second type of acetylcholine receptor, which is called the nicotinic receptor. So don't worry so much about the details there, under these diagrams. Just try to get the general idea. The nicotinic receptors are our second type of acetylcholine receptor, and they are ionotropic, meaning they act as an ion channel. So this is actually a good one to look at. If you look at this diagram on the right, I just circled a nicotinic receptor, and it acts as an ion channel. So essentially, acetylcholine binds to the receptor, it opens up an ion channel, and you get sodium moving in to the cell and potassium moving out. And this is actually the type of acetylcholine receptor we have at the neuromuscular junction. And it's excitatory because just remember back to muscles. It causes a depolarization and then an action potential. So it's stimulatory when acetylcholine binds. So these two types of receptors actually work very, very differently. Norepinephrine is released from pretty much all of the sympathetic postganglionic neurons. So that's really where you find norepinephrine. The axons that release norepinephrine are called adrenergic fibers, and the receptors for norepinephrine are called adrenergic receptors. So if you hear adrenergic receptor, it's one that binds to norepinephrine or epinephrine. Okay, so epinephrine can also bind to the adrenergic receptor. Alpha adrenergic receptors are one type of adrenergic receptor, but there are also receptors called beta adrenergic receptors. And one of the really interesting things about these beta adrenergic receptors is that they are clinical targets of what are called the beta blockers. And the beta blockers are actually what are called antagonists for the beta adrenergic receptor. So what they do is the beta blockers can bind to the beta adrenergic receptor, but they don't stimulate the second messenger system. They block it from taking place. So beta blockers are actually used as medications for control of high blood pressure. So what they can do is lower the amount of force that's generated in cardiac muscle by blocking the adrenergic signaling. So these are actually a really big class of hypertension treatment medications. So just to show you what we have here, this is norepinephrine, and these are actually the receptor types that norepinephrine can bind to. And you'll see these Gs that are kind of popping up. These are those G proteins that we were talking about way back at the beginning of the semester. Not every, norepinephrine effect is going to be excitatory. Some of them are inhibitory. And so if you see a G with an I by it, all of those are going to be kind of inhibitory pathways and have an inhibitory effect on cells. If you see a GQ or a GS, those are stimulatory pathways most of the time, and they will cause, kind of an excitatory effect on cells. But don't, feel the need to try and memorize any of that stuff. Just know that the different receptor types for norepinephrine are going to be potentially linked to different G proteins and therefore have different effects on the cell. This is a chart that shows us a lot of those different types of receptors. And, one of the things is if you do go into nursing or medicine, you're going to become very comfortable with charts like these. Because what you'll notice is that, it's got what are called agonists and antagonists listed. And I do want to touch on what those are a little bit more in this slide because clinically they're very important. The antagonists are what are called blockers. So what they can do is they can bind to a given receptor type and block it. So, we'll talk about a good example here with muscarinic receptors. So the M135 receptors are blocked by something called atropine. And this is actually used clinically. The M135 receptor is found on the heart, and there are people who have their blood pressure or heart rate drop to critically low levels for a variety of different reasons. They can have an overactive vagus nerve. It releases acetylcholine onto the heart, and what it'll do is it'll cause a drop in blood pressure and heart rate, and it can potentially be fatal. So if you can block the acetylcholine from binding to those M135 receptors, you can actually restore blood pressure. So if someone's blood pressure drops to critically low levels, a physician can give them atropine as a way to block the parasympathetic effect. And then they probably give, something that is an agonist or would stimulate, a sympathetic effect, because the sympathetic effect on blood pressure and heart rate is to increase it. So agonists are things that can stimulate the receptor, whereas the antagonists are ones that block it. Not every receptor, especially when it comes to the adrenergics, bind things in the same quantities. So all the adrenergics can bind norepinephrine and epinephrine, but sometimes, you'll have one receptor type that can bind one more than the other. So that's why you see norepinephrine is greater or equal to epinephrine. That just means that this alpha 1 receptor tends to bind more norepinephrine, or at least as much epinephrine. So just to kind of decode what some of these things mean. So don't really feel the need to memorize this chart in any way. But if you do go to nursing school, you're going to learn about a lot of these because you take a whole class called pharmacology, and this is a big part of it. A lot of these are used clinically, as a way to treat various medical conditions. The autonomic effects are sometimes not directly related to organ function, but rather a change in blood flow to the organ. So sometimes the autonomic effect would be to cause vasodilation. Vasodilation is when you open up blood vessels more and they can have more blood flow, and that can cause an increase in activity in that organ. Conversely, if you do vasoconstriction and you decrease blood flow to an organ because you narrow the blood vessel, that can decrease the activity of the organ. So you do see this with glands of the body that release different substances like saliva, for example, but you also see it with things like the kidneys. The kidneys are very sensitive to changes in blood flow. It directly impacts how they work. So it's not just glands that, are affected by the changes in blood flow, but it is one way to also change the function of an organ is by modifying how much blood goes to the organ. Also, sympathetic effects do tend to last longer than parasympathetic. And one thing that I want you to think back to is what chemical the parasympathetic nervous system uses. It uses acetylcholine. Does anyone remember how we get rid of acetylcholine in a synapse? There is an enzyme that breaks down acetylcholine. It is everybody's favorite enzyme, but maybe not to spell. It's acetylcholinesterase. Acetylcholinesterase breaks down acetylcholine in synapses very quickly. So we can shut off parasympathetic effects really quickly. We don't have an analogous chemical for norepinephrine. Norepinephrine actually has to be reabsorbed by the nerve. It will diffuse to different tissues, pass into the bloodstream, and then it has to be cleared by your kidneys via the urine. So I guess what I'm trying to say is, if you got a lot of release of norepinephrine or epinephrine, either one, it's going to take you at least 24 hours to clear it from your system. So that's why, if you did have an adrenaline rush or a panic attack, you're going to feel kind of on edge for a while because it takes so long to clear those chemicals from your system. Parasympathetic effects are shut down a lot faster because of the presence of acetylcholinesterates at your synapses. There are some substances that are released along with acetylcholine or norepinephrine, and they are these really interesting things called co-localized transmitters, or sometimes we just call them neuromodulators. So you may not just strictly release epinephrine or norepinephrine from a sympathetic postganglionic neuron. You could release one of these other chemicals too. And again, don't know all of them, but the ones that I specifically point out, of course, you would want to know. So let's say that you have a fight or flight stress response. Not good, right? But you release along with your epinephrine and norepinephrine something like an enkephalin. The enkephalins, if they're released along with epinephrine, norepinephrine, what they will do is have kind of a pain relieving effect. So think about this in terms of survival. Let's say, you got bit by a dog, right? And you were trying to get away from the dog. Well, what would happen is that you'd release norepinephrine and epinephrine into the bloodstream, but you'd also release enkephalin too. And the enkephalin will cause some pain relief so you can focus more on escaping rather than the excruciating pain that you're in. So the neuromodulators are actually pretty important for trying to help make the effect of the chemical like epinephrine or norepinephrine work better biologically. The parasympathetic nervous system doesn't have as many of those, though we do know of one called nitric oxide. It is a gas, and it triggers vasodilation of blood vessels by inhibiting the tone of smooth muscle. But most of the neuromodulators are found in the sympathetic nervous system. Okay, your organs are, generally speaking, duly innervated. Most of them are. It means that they have innervation from both the sympathetic and parasympathetic divisions. And most of the time, they have what is called an antagonistic effect. That just means opposite effects. So you guys already know this. The sympathetic nervous system will have a different effect on heart function than the parasympathetic nervous system. They're totally opposite effects. That is an antagonistic effect of the autonomic nervous system. Sometimes the two divisions have what is called a cooperative effect, means they can act on different effectors to produce a unified effect. And we'll take a look at an example of that in a minute. A good, just to know off the bat, example of a cooperative effect is production of saliva. The sympathetic and parasympathetic divisions actually both are involved in saliva production. You should know that the two divisions don't necessarily innervate an organ equally. There is way more extensive innervation by the parasympathetic division of digestive organs. And it makes sense. That's the rest and digest division of the autonomic nervous system. So of course there's going to be more extensive wiring to a digestive organ by the parasympathetic nervous system. The sympathetic division is going to have greater innervation and effect on the muscle in the heart. The parasympathetic nervous system actually doesn't really innervate the muscle in the heart. It only innervates the pacemaker and can change the heart rate. of the heart. But the sympathetic division is because it can innervate the pacemaker of the heart and the muscle of the heart, it can affect not only the pacing or the heart rate, it can affect the force of how hard the heart contracts, which is going to change blood pressure too. This slide is giving us a little bit more information about the antagonistic and the cooperative effects that the autonomic nervous system's two divisions can have on structures of the body. The antagonistic effects can happen in one of two ways. So the first way that it can happen is that by having dual innervation, the effect of the chemicals or the receptor types can cause different effects on the same organ. So the acetylcholine from the parasympathetic division and the norepinephrine from the sympathetic division, when they act on the heart cells, they act in the same heart cells but have very different effects. So for the parasympathetic division, the acetylcholine would decrease the heart rate by essentially decreasing the activity of the pacemaker cells, whereas the sympathetic division will work on those same cells, but through the actions of norepinephrine will stimulate their activity level. So in this first scenario, we're actually, the chemicals are working on the same effector cells, we just have opposite functions. You can also have antagonistic effects because a division innervates different cells. And this is actually what you see in the eye. In the eye, the sympathetic nervous system stimulates the contraction of a muscle called the pupillary dilator muscle. And that triggers dilation of the pupil, which will increase the amount of light coming into the eyeball, which is important in the fight or flight stress response. You can get more visual information about your surroundings. The parasympathetic division actually innervates a different part of the eye. It innervates a muscle called the constrictor pupillae muscle. And when that muscle is contracted by the parasympathetic nervous system, it constricts the pupil and limits the amount of light going into the eye. So it is a different effect by those two divisions, but it's because we're impacting different structures within that same organ. Cooperative effects are when the two divisions work together to produce a unified effect. And a good example is saliva production. The parasympathetic division will actually cause an increase in the production of one part of the saliva that is really thin, slippery, and enzyme rich. It's called serous fluid. The sympathetic division produces saliva as well by essentially increasing the production of mucus. And mucus and serous fluid are both part of saliva. And again, that's something that you learn more about in A&P2, but they both work together to produce saliva, just different parts of the saliva. This is just a picture showing us the antagonistic effects because each division innervates different cells. So that's showing us the pupillary dilator muscle and the constrictor pupillae muscle. Okay. There is control of body structures without dual innervation sometimes. So what that means is you might have a body structure that's only controlled by the sympathetic nervous system or only controlled by the parasympathetic nervous system. These are some body structures that are only innervated by the sympathetic nervous system. The adrenal medulla, which is the inside of the adrenal gland, the erector pili muscles in the skin, sweat glands, and actually most blood vessels are only innervated by the sympathetic nervous system. So keep that in mind. I think, it is important to emphasize this point a little bit more. Most of the blood vessels are controlled only by the sympathetic nervous system. And the sympathetic nervous system helps maintain the tone of those blood vessels using something called sympathetic vasomotor tone. Essentially, even at rest, the sympathetic nervous system has a baseline firing frequency, basically a low level of activity that helps keep your blood vessels in a partial constriction state. And this is actually pretty important for just helping you maintain normal blood pressure. Now, if you did have, let's say, a fight or flight stress response, the sympathetic nervous system would become more active by increasing the firing frequency. So it sends epinephrine, norepinephrine more frequently, which can lead to vasoconstriction, and that actually raises blood pressure. It can also, in times of, relaxation mode, a decrease in the firing frequency of the sympathetic nervous system would cause vasodilation, and that's actually a mechanism to lower blood pressure. The idea behind that is this, guys. Let's say you had a fight or flight stress response. You got bit by a bear and you were losing blood. Okay, bad situation all around. Your sympathetic nervous system would fire like crazy because it's your fight or flight system, the body. That's going to trigger vasoconstriction, especially of blood vessels in the skin. But what that'll do is a few things. It's going to divert blood to important structures like the heart and the muscle. If you get bit by a bear, you need to fuel your muscles and your brain and your heart so you can escape that stressor. And by diverting blood to those locations, you're going to move blood away from the surface of the skin. So those blood vessels near the skin constrict, and that's actually going to help you limit blood loss too. So this is a very sophisticated mechanism used by the sympathetic nervous system to not just help maintain normal blood pressure, but also help you adapt to a stressor, allowing you to shuttle blood to where it really needs to be when it's needed there, as well as potentially limit blood loss in the event of, a vascular injury that could cause you to lose blood. So the sympathetic division does prioritize blood vessels that flow to muscles and the heart and also the brain in times of stress. And just the idea behind that is to shuttle blood to where it needs to go, which are absolutely the muscles, the brain, and the heart in times of an emergency, as well as to limit the blood flow if there is an emergency situation and a vascular injury. So it's a very important mechanism to help deal with those emergency situations. The autonomic nervous system is a motor division, right? So this is a motor division, all right, visceral motor, so we remember. And that means that it is part of the peripheral nervous system, right? But the peripheral nervous system is controlled by the central nervous system. The brain and spinal cord control everything, right? So we do have control of the autonomic nervous system by the central nervous system. And these are just some of the key areas of the central nervous system that regulate autonomic function. The cerebral cortex is involved in higher brain functions. The limbic system is involved in emotional output for the brain, and it can send that information to the hypothalamus, which is an autonomic integrator. It is the main integrator of autonomic information, and what it can do is it can direct the stimulation of either the parasympathetic or sympathetic division if the time is appropriate. So if you had something, sad happen, it kind of upsets you, and your limbic system detected that, it could send that information to the hypothalamus, so-and-so is upset, let's increase heart rate and breathing patterns. And so what the hypothalamus will do is it will use interneurons that stretch from the hypothalamus down to the spinal cord and essentially cause activation of the sympathetic nervous system. And so we kick off the chain in the thoracolumbar spinal cord that would cause you to stimulate your heart, beat it a little faster and a little bit harder, and also make your lungs breathe a little bit more frequently as well. So remember, there is central control of autonomic function. It's not just that sympathetic and parasympathetic divisions always know what to do by themselves. They're taking orders from the central nervous system, so they know the appropriate way to change organ function. And that's really the big idea here. This is just a chart showing us some of those autonomic effects in organs, and you don't need to know all of them, but you should know, I would say know 3 effects on 3 different organs for the sympathetic and the parasympathetic divisions. We've already done some of them, so we've already talked about like the sympathetic division, dilating the pupils, relaxing the airways to bring more air into the lungs, accelerating and strengthening heartbeat, and some of these other ones as well. Keep in mind that the sympathetic nervous system tends to stimulate things like the heart and the lungs, but shut off things like digestion and other non-essential biological activities. So sympathetic division is big on prioritizing survival mechanisms. So the parasympathetic division will have generally contrasting or opposite effects on those organs. It'll slow heart rates, but stimulate digestion. It's rest and digest, right? That's what it's called. So it will do things like stimulate digestion or urine production or those type of activities. That brings us to the end of the material for this semester. Please let me know if you have any questions about this unit. It is a challenging one to end the semester with, and it is definitely expected that you will have some questions as you process and learn this information. So please feel free to reach out to me and let me know if you have any questions. I also wanted to say great work this semester. I'm so proud of each of you and how responsible and accountable for your academic work you've all been this semester. I know it's a challenge sometimes to do these sort of classes online, but I think you've all risen to the occasion and done a great job. So great work.